Hello, Secondary 1 students. Our biology lesson today is 6 linked, 6 influenced, and 6 limited traits. All of our lesson is talking about non-Mendelian characters. Mendelian characters, as we studied before, related to a gene which dominates over the other. This means that there is a dominant gene and there is a recessive gene, but non-Mendelian characters are different. They are 6 linked traits, 6 influenced traits, and 6 limited traits. Let's start with the first one, 6 linked traits. From examples of 6 linked traits, hemophilia disease means blood liquidity or means absence of some clotting factors which causes clotting of the blood. And color blindness, which causes the eye not to be able to distinguish or differentiate between certain colors, especially the red and green colors. Some other diseases like muscle atrophy and some somatic characters like dominates of the red color eye in Drosophila insect over the white eye colors in Drosophila insect also. The scientist Thomas Morgan studied this character 6 linked traits in Drosophila insect or fruit fly insect. He discovered that the gene that causes the eyes to be red is located on the 6th chromosome, not on the somatic chromosomes. So he named them 6 linked traits. Let's study it. Okay, here we have the 6 chromosomes, X and X, so it appears that it's a female. The rest of chromosome carry the somatic characters like the eye color and the length of the wings and so on. And the other part of the chromosome will carry the 6 characters which appear on the 6 of the fly. On the other side, here are the 6 chromosomes of the male, X and Y. As we said before, that Y will be shorter than X chromosome. The rest of chromosome X carry the same characters on the other side, but this part will carry some of the somatic characters, some of the characters that will appear on the Drosophila fly body from outside that can we distinguish it by our eyes. But as we see here, the Y is a short chromosome that missing this part, so this character will not be loaded or will not be carried on the Y chromosome. So this character will be carried on X chromosome only. So sex-linked traits are somatic traits, means its character will appear on the body, that their genes are located on 6 chromosomes. This character will be carried on the 6 chromosomes but not affected by the sex, and their appearance not affected by the sex hormones or the sex of the living organism. Example, as we said before, the eye color in Drosophila insect, color blindness, hemophilia, blood liquidity, short-sightedness, and muscle atrophy in human. Here we have some genotypes to study to can differentiate between the different genotypes. X and X, it appears that it's a female. So what are R capital and R capital? These are the genes which represent the somatic characters which are carried on the 6 chromosome. When the female carry R capital and R capital, so its eye color will be red and it's a pure. X and X, so it's also female but carry R capital or small, but the R capital represents the eye color, the red eye color, which is dominates over the white, so this female is a red-eyed color. XY means this insect is a male, its genotype is a male, 6 chromosome, and the R capital represent the gene, the somatic chromosome or the gene which will produce the red color eye. And XY it's a male, but R small is a recessive gene So this male will be a white colored eye. So this female is red, this female is red colored eye, this male is red colored eye, and this male is white colored eye. Let's take example here. Okay, we have a male here. So this gene is X and this gene or chromosome is Y. So this chromosome is X and this chromosome is Y. The curved one is Y. And the female here, XX. So we will write it XX. Here is the sex chromosome. So where are the genes that are carried on the sex chromosomes? This male is white-eyed color. So it will take R small on the X chromosome. We said that Y chromosome will not carry this character because it's short and missing the part which carry this character. And the female here is XX and red colors. So she will has R capital here and R capital on the other chromosome. Let's take the gamete. They are similar chromosomes and they carry the similar genes, so it's enough to take one of the chromosomes because they are similar. XR capital. On the other side, let's take the gametes of the male. X, which carry the recessive character, are small, and Y will not carry anything. After making a crossing between these gametes, XR capital and XR small, so this female will be produced X, which carry the R capital, the red colored eye gene, and X, which carry the R small of the white color. On the other side, XR capital will make a fertilization to Y, so it will produce this male XR capital and Y small. Okay, takes the gametes of the first generation individuals, XR capital and XR small, here XR capital with Y, which will not carry anything. The first generation produces female with a red color and male with a red color also. When crossing occurs between them, so this gamete with this gamete, R capital produce this female, R capital, R capital. It's a female with a red color. When crossing occurs between XR capital and Y, so will produce this female, XR capital. Sorry, here XR capital and Y, this male, R capital and Y. This male will be red-eyed color. XR small with XR capital here will produce this female, XR capital and XR small, and the last possibility of fertilization between this gamete and this gamete, XR small and Y, XR small and Y. As we can see here that this female is red-eyed color, this female is red-eyed color, this male is red-eyed color, and this male is white-eyed color. Okay. This male carry the recessive gene, which is R small, which will produce the white-eye color from where? From this chromosome. Okay, and this chromosome coming from where? From this male. So every grandson can inherit the character from the grandfather, and this gene of grandfather is carried or inherited through his mother, because the grandfather, inherit this character to his daughter, and his daughter transfer this character to grandson. Let's represent this by another shape. So if we have XX R small, so this female is wide-eyed because she carries R small and R small. On the other side, the male XY carry R capital, so it's red-eyed color. Takes the gamete, segregate the gametes, and make a crossing between them. XR small with XR capital will produce female with a red-eyed color because R capital dominates over R small. And XR small with Y will produce male which carries the R small, so it will be white-eyed color. If we're making a cross between these two individuals which resulted from the first generation, so takes the gamete, as we studied before, and make a crossing between them, so the result will be female with a red color, female with a white color, male with a red color, and male with a white colored eye. Okay. Morgan can consider the eye color character in Drosophila as number 1, Mendelian character. Why? Because 100% of the first generation appears with the dominant character, which is red-eyed color. And when a crossing occurs between them, the second generation will be 3 dominant and 1 recessive. 3 contains the red-eyed color or carry the red-eyed color, and 1 of the 4 individuals will carry the recessive character, which is white-colored eye. If we consider that we are talking about the phenotype or the color instead of the sex of the insect. He can consider this character also a sex-linked character, as quarter of the second generation will carry the white colored eye or the recessive colors, and they are males. So the sex here determines that the character transfers from the grandparents through their daughters, then to the grandsons. Okay, the gene carried on the sex chromosome X, but Y carries a few of them, or will not carry at all. Let's take example in a human. We talked about the eye color in Drosophila insect, and we will transfer to the sex-linked characters in the human. Let's start with the color blindness in human. In the color blindness, the patient can't distinguish between different colors, especially the red and green colors. Okay, here is a test. Test if you can distinguish between the red and green color. If you passed it, you are not infected with the color blindness. The color blindness gene is a recessive gene, means it's a small letter which is carried on the X chromosome. As we said before, that Y will not carry anything. Here if the crossing occurs between this mother, which is XX, and carry C capital and C small. This C small gene, if it transfers to the any of the daughters or sons, will infect them. Okay, and here is the genotype of the male or the father, XY, and X is a capital, so it's not infected with any disease. He's not has the color blindness. So crossing occurs between gametes X capital, so this female will not be infected. If occurs between X C capital and Y, so will produce this individual. As long as it carries C capital, is not infected. Okay, the crossing occurs between this gamete XC small and XC capital will produce hybrid female C capital C small, so she's a carrier, not infected woman. X C small and Y, this is will carry the recessive gene, so this male will be infected or has a color blindness disease. Another example of the sex-linked traits in human is a hemophilia. Hemophilia or blood liquidity means that the blood lacking the clotting factors responsible for making the blood clot in case of injury occurs due to lack of some substance, as we said before, caused by recessive gene also in X chromosome, and repeat again, Y chromosome will not carry this character, and this may lead to death. Okay, let's make a cross between female XX carry the H small. H small represent the gene which will infect the person with blood hemophilia, and a male XY which carries the H small. Making a cross between gametes, XX will carry the H small, so this female will be infected with blood hemophilia. XY will not carry any genes, so this man will not be infected with any disease. XX, both of them contains H small, and H small is the recessive gene which carry the blood hemophilia disease, so this female for sure infected with blood hemophilia disease, and XY here carry the H small, so this male will be infected with blood hemophilia disease. From the previous, we can conclude that these characters, sex-linked characters, are dominant in males than in females. Why it dominates in males than females? Wait a second. Why this character dominates in males than in females? Here, because in males, it represents by one gene only. In males, it represents by one gene only. One gene on what? On X chromosome, as Y doesn't carry the gene. But in females, it represents by a pair of genes. Pair of genes because the female will carry X chromosome, and each one of them has a possibility to carry this gene. But in males, it represents by only one gene, which is carried on X chromosome, not on Y chromosome. Okay, number 2, we can conclude that male passes the gene of trait to his daughters and doesn't pass it to his sons. Why? Because he passes Y chromosome to the males only, and the chromosome X which carries the gene of trait to the females or daughters only. Okay, so the male will pass the gene to the daughters because the male XY will carry the gene on X. So who will take the X? The females, but Y will transfer to his sons, so his sons will not carry any character. But the carrier mother who takes the X chromosome from the father will transfer it to her daughters or her sons. So the female can transfer the character, the female which is carrier will transfer this character or this gene from her father to her daughters or her sons, both of them. She can carry the character to both her daughters or her sons. So the male will pass the gene of trait to his grandsons through his daughters, which are carriers. The mother, as we said before, passes the gene of trait to her sons and daughters, and sons will inherit the trait from their mothers, not from their fathers, whereas the trait appears on daughters when they inherit the gene of trait from both their father and mother. Let's transfer to the second non-Mendelian character, which is sex-influenced trait. Here, it's completely different. The genes will not be carried on the sex chromosomes on X or on Y, but it will be affected by the sex hormones, affected of being this is male or female, where the hormones that are secreted on maturity from the gonads of male and female will affect on this character. It will not appear before maturity. It will appear after maturity. From examples, genetic baldness trait in human, like that, and presence of horns traits in cattle. But the man will be infected with a hair loss, with a baldness, but in females will not affected by baldness, she will be affected by hair losing. So the sex influence traits when presence of female or male hormones influence expression of a certain human traits. Example of the baldness, as we said that it's a somatic chromosome, so it will take a letter B, baldness, and this plus will represent the amount of hormone that will affect on our trait, the amount of hormones secreted on puberty that will affect on the trait. So if the male has a genotype BB and has 2 plus, so it will be infected by baldness. If the female had these 2 plus signs, so she will be affected by losing hair. If the male has a genotype B capital and has only 1 plus sign, so he will be affected because this amount of hormone, testosterone or masculinity hormone, which is secreted from the male only, is enough to express this trait. But in female, 1 plus is not enough to express this trait, so it will be a healthy female. If the female contains B and 1 plus, as we said, that she doesn't affected by losing hair. Let's take an example. B capital will be in both sexes, in males and females, because it's represent the somatic trait. But look at the plus signs. Here, B capital and 1 plus represents a woman with a normal hair, because 1 plus only not enough to express this trait. And if we talk about the man, 1 plus is enough to express this trait and causing baldness for this man. Takes the gametes and making a crossing between them, as we learned before. So the gametes will be make a fertilization with each other. So the first generation will be genotype B capital with 2 plus, B capital with 1 plus, B capital with 1 plus, and B capital with no plus. Here, if we are talking about the males, 2 plus making baldness, 1 plus making baldness, 1 plus making baldness. So all of these 3 genotypes, if they are males, they are affected by baldness, and this is a normal male because there is no any genes here represent this trait. But if we are talking about the females, 2 plus is enough to making a hair loss, 1 plus is not enough, 1 plus is not enough, and there is no plus here, so she's a normal hair female. The 3 genotypes are normal hair female, but this is a 1 hair falling female. So the genotype here is different from males and females. So the result here, we can distinguish between males and females. We can say that all of the 3 males are affected by baldness. All of the 3 females, the last 3 females, are not affected by hair loss. Here's a summary between the 6 linked traits and 6 influenced traits. You can read them again to summarize what we already explained. The last character or the last non-Mendelian character is the 6 limited traits. Sex-limited means that traits which appear in only one of the two sexes, restricted, or will not appear in the two sexes, it will appear in only one sex, according whether it's male or female. Like what? Like the milk production. Milk production is restricted to the females only. Like beard, it's restricted to the men only. Like the ability of the birds to lay eggs, it's restricted to the chicken only. What about the medical tests that done before marriage? It's a group of medical tests that are carried out by the couple, the male and female, before marriage. These are searching for some diseases like AIDS, the immunity deficiency syndrome, or like thalassemia, which is a type of anemia, and so on, to make a choice or advice to the couple in order to help them to make a healthy family. And what makes genetic disease spreading? Relative marriage and getting married without carrying out medical tests, like as we said before. What is the importance of medical tests? Medical tests help in having healthy children. If we discover some disease, we can prevent it. To limit the spreading of genetic disease, congenital disorders, and mental retardation, and avoiding social, theological, and financial problems resulted from taking care of children with genetic disease. We can avoid all of these mistakes or all of this headache that resulted from genetic disease that occurs if we didn't do this tests. DNA fingerprint. What's DNA fingerprint? DNA fingerprint is the arrangement of nucleotides or certain repetition of DNA segments inside the body. So it's unique for everyone, especially, exception of the identical twins. The identical twins may have the same DNA fingerprint. So to recognize a person by a comparison of his DNA sequence with another, we make a certain analysis called DNA fingerprint. What is the importance of DNA fingerprint? It's important in the field of medicine to study the genetic disease. So when we separate the segments of DNA, we can identify a certain gene which is responsible for a certain disease and used in tissue transplantation process. When we decide to transfer a certain organ like a liver or like a kidney to other person, there is some medical diagnosis or some medical analysis done to be sure of matching between the blood groups and matching between the DNA of the patient and of the donor. Other field using the DNA fingerprint, field of forensics, to recognize the deformed dead bodies and tracking lost children and help the courts begin investigations in crimes. It also will discover the criminal. It plays important role also in parentage cases. What is the human genome? I think that we talked about it in the beginning of the term. The human genome contains all the genes exist in the nucleus of somatic cell. And what are their number? How many? They are from 60,000 to 80,000 genes in the nucleus. They are carried on 23 pairs of chromosomes. So the summation of all the genes that found in one somatic cell, they are called the genome, human genome. Watson and Crick scientists discovered that the genes are carried on the double helix DNA. Okay, and this is the history of the discovering the genes number. And what are the importance of the genome? Scientists aim to use genome in what? In manufacture of drugs without side effects, in studying evolution of the living organism by comparing the human genome to that of other organisms and improving offspring by determining the genes causing disease in the fetus before its birth and improving them. Thank you for listening.